Hello everybody, myself Padma and today we will be discussing BHU PET 2019 MSc Plant Biotechnology question paper. Coming to the first question, the interactions between the non-polar molecule in a polar solvent are called option 1 hydrophobic interaction, 2 hydrophilic interactions, 3 van der Waals interactions, 4 electrovalent interaction. Answer is option 1 hydrophobic interactions. Next one, which form of DNA has the longest pitch? Option 1 bDNA, 2 aDNA, 3 cDNA, 4 zDNA. Answer is option 4 Z. Next one, most of the biological oxidation occur without direct participation of oxygen. Option 1 true, 2 false, 3 no process like biological oxidation, 4 in biological system only chemical reduction occur. Answer is option 1 true. Next one, the role of Recce protein is option 1 homologous recombination, 2 initiation of DNA replication, 3 synthesis of lagging strand, 4 increase the processivity of DNA polymerase 3. Answer is option 1 homologous recombination. Next one, the DNA polymerase 3 has dash exonuclease activities. Option 1 3' to 5', 2 5' to 3', 3 3' to 3', 4 5' to 5'. Answer is option 1 3' to 5'. Next one, assume two conditions. 1 crossing over present, 2 absent. During cell division, the two alleles of a gene will segregate during dash and dash respectively. Answer is option 2 anaphase 1 and anaphase 2. Next one, examples of hexaploid crops or species are option 1 bread wheat chrysanthemum triticale oat, option 2 bread wheat sugarcane strawberry oat, 3 bread wheat chrysanthemum strawberry oat, and 4 bread wheat cotton peanut triticale. Answer is option 1 bread wheat chrysanthemum triticale and oat. Next one, in genetic engineering a chimera is option 1 a plasmid that contains foreign DNA, 2 an enzyme that links DNA molecules, 3 a virus that infects bacteria, and 4 a fungi. Answer is option 1 a plasmid that contains foreign DNA. Next one, gene for gene hypothesis states that answer is option 2 for each resistance gene in the host there is a corresponding gene for a virulence in the pathogen conferring resistance and vice versa. Next one, assembling multiple desirable genes from multiple parents into a single genotype for specific trait is known as option 1 multiple resistance, 2 multi-line variety, 3 gene interaction, 4 gene pyramiding. Answer is option 4 gene pyramiding. Next one, nuclear polyhedrosis virus is a major disease of option 1 silkworm, 2 aphids, 3 jacid, 4 maggot. Answer is option 1 silkworm. Next one, which of the following is a major pest of paddy? Infescutilus, 2 Stylophilus oryza, 3 Neelaparvata ligens, and 4 Athrigona saccata. Answer is option 3 Neelaparvata ligens. Next one, pheromones are synthesized by option 1 thoracic gland, 2 neurosecretory cells, 3 glandular epidermal cells, and 4 all of these. Answer is option 3 glandular epidermal cells. Next one, molting hormone is secreted by option 1 corpora ellata, 2 brain, 3 neurosecretory cell, 4 prothoracic gland. Answer is option 4 prothoracic gland. Next one, cultivation of lathyrus in paddy fields is an example of option 1 multiple cropping, 2 relay cropping, 3 intercropping, 4 mixed cropping. Answer is option 2 relay cropping. Next one, the food or fortified food product that not only supplements the diet but also assists in treating or preventing diseases apart from anemia, so provides medical benefits. Such products are known as option 1 pharmaceuticals, 2 ultraceuticals, 3 cosmeceuticals, and 4 nutraceuticals. Answer is option 4 nutraceuticals. In honeybees, the males are options 1 XX, 2 XY, 3 haploid, 4 tetraploid. Answer is option 3 haploid. Next one, XYZ system of hybrid production is done in which crop? Option 1 rice, 2 wheat, 3 maize, 4 sorghum. Answer is option 2 wheat. Next one, the process of transfer of useful genes from exotic or wild types into agronomically acceptable background is called as option 1 recurrent selection, 2 reverse genetics, 3 pre-breeding, 4 transaggressive breeding. Answer is option 3 pre-breeding. Next one, Ratna variety of mango is developed from the cross of option 1 Neelam and Alfonso, option 2 Alfonso and Neelam, 3 Neelam and Dashehri, and 4 Dashehri and Neelam. Answer is option 2 Alfonso and Neelam. Next one, which of the following is non-climatic fruit? Option 1 mango, 2 apple, 3 banana, 4 lychee. Answer is option 4 lychee. Fruit of coconut is option 1 berry, 2 drupe, 3 nut, 4 pome. Answer is option 2. Next one, gladiolus is commercially propagated by option 1 corms, 2 bulbs, 3 stem cuttings, 4 rhizomes. Answer is option 1 corms. Pungency in radish is due to option 1 solanin, 2 capsaicin, 3 isothiocyanate, and 4 calcium acetate. Answer is option 3 isothiocyanate. Next one, mycorrhiza is an association between option 1 bacterial and fungi, 2 algae and roots of higher plants, 3 fungi and roots of higher plants, 4 none of these. Answer is option 3 fungi and roots of higher plants. Next one, denitrification is more in option 1 well drained soils, 2 waterlogged soils, 3 heavy soils, 4 light soils. Answer is option 2 waterlogged soils. Next one, generally in plant cell the mitochondria has the dash pH, cytosol has the dash pH, and lysosomes has dash pH. Answer is option 1 8, 7.2, 4.7, 2, 7.2, 8, 4.7, 3, 4.7, 7.2 and 8, and 4, 4.7, 8 and 7.2. Answer is option 1 8, 7.2 and 4.7. Next one, the Indian Seed Act was enacted in which year? Option 1 1966, option 2 1967, option 3 1968, and option 4 1969. Answer is option 1 1966. Next one, golden yellow color of tag is representative of which class of seed? Option 1 foundation seed, 2 certified seed, 3 registered seed, 4 breeder seed. Answer is option 4 breeder seed. Next one, in certified seed, the minimum standard for germination percentage of soybean is option 1 70%, 2 80%, 3 90%, 4 60%. Answer is option 1 70. Next one, minimum isolation distance in meters in hybrid bajra for foundation seed. Option 1 500 meter, 2000 meter, 3500 meters, 4 2000 meters. Answer is option 2 1000 meters. Next one, particle size analysis in a soil sample is performed based on the principle of option 1 Stokes law, 2 Laplace equation, 3 Bray law, 4 Atterberg's principle. Answer is option 4 Atterberg's principle. Next one, formation of well-defined ephemeral channels caused by slit-laden runoff is manifestation of option 1 gully erosion, 2 rill erosion, 3 sheet erosion, 4 stream bank erosion. Answer is option 1 gully erosion. Next one, the classical book Origin of Mendelism was written by option 1 Carl Correns, 2 R.C. Olby, 3 T.H. Morgan, 4 Hugo de Virus. Answer is option 2 R.C. Olby. Next one. Cyanobacteria is option 1 Azolla, 2 Azospirillum, 3 blue-green alga, 4 Rhizobium. Answer is option 3. Next one. If the values of set are measured in centimeters, the unit of variance will be option 1 no unit, 2 centimeters, 3 centimeter square, 4 centimeter cube. Answer is option 1 no unit. Next one. If all observed frequencies of class are same as expected, the value of chi-square is option 1, 1, 2, infinity, 3, 0, 4, none of these. Answer is option 3, 0. Next one. Homogeneity of several variances can be tested by option 1, Bartlett's test, 2, Fisher's exact test, 3, F test, 4, T test. Answer is option 1, Bartlett's test. Next one. Mendel did not observe linkage because option 1, synapses, 2, independent assortment, 3, mutation, 4, crossing over. Answer is option 2, independent assortment. Next one. Barney's burr star system is known for option 1, male sterility, 2, self-incompatibility, 3, open pollination, 4, self-pollination. Answer is option 1, male sterility. Next one. Which of the following chemical enhances Vir gene expression? Option 1, cyanidin, 2, glutenin, 3, acetosyringeon, 4, dextran. Answer is option 3, acetosyringeon. Next one. Under in vitro propagation method, bipolar development of plant initiation is called as option 1, organogenesis, 2, embryogenesis, 3, calogenesis, 4, androgenesis. Answer is option 2, embryogenesis. Next one. A satellite DNA is one which is option 1, present in satellite chromosome, 2, present in B chromosome, 3, very large arrays of tandem repeating DNA, 4, usually present in euchromatin region. Answer is option 3, very large arrays of tandemly repeating DNA. Next one. RNAi is used for option 1, gene silencing, 2, overexpression of gene, 3, transcriptional inactivation, 4, removal of introns. Answer is option 4. Next one. In the gametophytic self-incompatibility, the S locus products are option 1, synthesized after the completion of meiosis, 2, synthesized before the completion of meiosis, 3, synthesized before the flower development in sporophytic cells, 4, synthesized after double fertilization. Answer is option 1, synthesized after the completion of meiosis. Next one. In the agrobacterium mediated tDNA gene transfer, the foreign gene is inserted in tDNA because option 1, it naturally has multiple cloning sites, 2, it carries gene required for virulence, 3, the tDNA has flanking sequences which help in integrating in host DNA, 4, it has multiple origin of replication in 2 different organisms. Answer is option 3, the tDNA has flanking sequences which help in integrating in host DNA. Next one. Set of closely related genes or genetic markers that are inherited as a single unit is option 1, cistron, 2, gene families, 3, haplotype, 4, haploid. Answer is option 3, haplotype. Next one. The development of embryo from male gamete without fertilization is known as option 1, androgenic haploids, 2, gynogenic haploids, 3, apospory, 4, apogamy. Answer is option 1, androgenic haploids. Next one. The majority of DNA synthesis occurs during dash phase of cell cycle. Option 1, S, 2, G1, 3, G2, 4, M. Answer is option 1, S. The next one. The number of chromosomes in rice is option 1, 14, 2, 18, 3, 24, 4, 30. Answer is option 3, 24. Next one. The number of chromosomes 2n in wheat triticum durum is option 1, 14, 2, 28, 3, 42, 4, 56. Answer is option 2, 28. Next one. Commercially grown single cross hybrids are option 1, homozygous and homogeneous, 2, heterozygous and homogeneous, 3, homozygous and heterogeneous, 4. Heterozygous and heterogeneous. Answer is option 2. Heterozygous and homologous. Next one. Polycistronic mRNA is generally produced in option 1. Eukaryotes, 2. Both prokaryotes and eukaryotes, 3. Prokaryotes, 4. Neither in prokaryote nor eukaryote. Answer is option 3. Prokaryotes. Next one. Dash is a sequence in dash where RNA polymerase initially binds. Option 1. Promoter, DNA, 2. Promoter, RNA, 3. Enhancer, DNA, 4. Enhancer, RNA. Answer is option 1. Promoter, DNA. Next one. In DNA, the linkage between sugar and base is known as dash bond. Option 1. Phosphodiester bond, 2. Hydrogen bond, 3. N-glycoside, 4. Ester. Answer is option 3. N-glycosidic bond. Next one. The RNA primer during replication is synthesized by dash enzyme. Option 1. RNA polymerase, 2. RNA primase, 3. DNA polymerase, 4. DNA primase. Answer is option 2. RNA primase. Next one. The role of I gene in lac operon is option 1. Synthesis of lactose, 2. Synthesis of inducer, 3. Synthesis of repressor, 4. Synthesis of allolactose. Answer is option 3. Synthesis of repressor. Next one. Meiosis is important for option 1. Growth, 2. Repair, 3. Reproduction, 4. Disease resistance. Answer is option 3. Reproduction. Next one. In callus culture, option 1. Increasing the levels of cytokinin to the callus induces shoot formation and increasing level of auxin promote root formation. Next one. Increasing level of auxin to callus induces shoot formation and increasing level of cytokinin promote root formation. Next one. Auxin and cytokinins are not required, and 4. Only auxin is required for root and shoot formation. Answer is option 1. Increasing level of cytokinin to a callus induces shoot formation and increasing level of auxin formation. Next one. Bordeaux mixture is option 1. Mercury fungicide, 2. Copper fungicide, 3. Sulfur fungicide, 4. None of these. Answer is option 2. Copper fungicide. Next one. Little leaf of brinjal is caused by option 1. Bacteria, 2. Virus, 3. Phytoplasma and 4. Nematode. Answer is option 3. Phytoplasma. Next one. Synthetic mRNA AGA, GAG, AGAG produces polypeptide which incorporated arginine and glutamate. AAG, 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 AAG produces polypeptide which incorporated arginine along with other amino acid. The code for arginine is option 1. GAG, 2. AAG, 3. AGA, 4. GAA. Answer is option 3. AGA. Next one. How can we get haploid plants? Option 1. Organ culture, 2. Embryo culture, 3. Anther culture, 4. Tissue culture. Answer is option 3. Next one. The first nucleotide of nascent mRNA has dash phosphates. Option 1, 1, 2, 2, 3, 3 and 4, 4. Answer is option 3, 3. Next one. The amino acid is linked to tRNA by dash bond. Option 1. Ester bond, 2. Phosphodiester bond, 3. Glycosidic bond, 4. Peptide bond. Answer is option 1. Next one. DNA mitochondria is generally option 1. Linear, 2. Single copy, 3. Single stranded, 4. Circular. Answer is option 4. Circular. Next one. Who identified the killing effect of bacteria Bacillus thuringiensis on a Mediterranean floor moth in 1911? Option 1. Michael Faraday, 2. Enrico Fermi, 3. James Clerk and 4. Ernest Berliner. Answer is option 4. Ernest. Next one. The cultivation of vegetables in soil-less aqueous medium is called option 1. Molecular farming, 2. Tissue culture, 3. Hydroponics, 4. Mariculture. Answer is option 3. Hydroponics. Next one. In general, biologically active form of abscisic acid is option 1. Cis-abscisic acid, 2. Trans-abscisic acid, 3. Alpha-abscisic acid, 4. Beta-abscisic acid. Answer is option 1. Cis-abscisic acid. Next one. Which gene transfer technique involves the use of a fatty bubble to carry a gene into a somatic cell? Option 1 bialistic method, 2 liposome mediated, 3 electroporation, 4 in situ injection. Answer is option 2 liposome mediated. Next one. If one cell has AT content 40%, what will be the percentage of guanine residue? Answer options are option 1 60%, 2 15%, 3 30%, 4 guanine residue cannot be calculated. So they have given 80 percentage as 40. So the percentage of GC will be 60. So out of this, percentage of G will be 30%. Answer is option 3, 30. Next one. Cesium chloride will separate DNA molecule on the basis of option 1 absorption, 2 resorption, 3 density, 4 adhesion. Answer is option 3 density. Next one. In the study one experiment, it was found that the value of Tm for DNA is 40 degrees Celsius. If the cell has 20% GC at the above Tm, then what will be the value of Tm if the G percent increases to 60%? Tm increases with increasing percentage of G plus C. So answer is option 2 increases. Next one. Which of the following is not relevant to recombinant DNA safety guidelines in India? Option 1 IBSC, 2 RCGM, 3 GEAC, 4 NBPGR. Answer is option 4 NBPGR. Next one. The process of finding relative position of gene on the chromosome. Option 1 gene tracing, 2 gene mapping, 3 gene walking, 4 chromosome mapping. Answer is option 2. Next one. Proteins isolated from bacteria that catalyze specific cleavage of DNA are option 1 restriction enzyme, 2 bacteriophage enzyme, 3 methylating enzyme, 4 telomerases. Answer is option 1 restriction enzyme. Next one. A small circular DNA molecule used as a vector to transmit foreign DNA is A. Option 1 plasmid, 2 prion, 3 liposome, 4 lipofection. Answer is option 1 plasmid. Next one. The TI plasmid is generally used to genetically engineer option 1 bacteria, 2 plants, 3 fungi, 4 animals. Answer is option 2 plants. Next one. Which of the following enzymes can be described as a DNA dependent RNA polymerase? Option 1 primase, 2 DNA polymerase, 3 DNA polymerase 1 and 4 reverse transcriptase. Answer is RNA polymerase, but it is not given in the option, but they have given primase, which is also a type of RNA polymerase. So answer is option 1 primase. Next one. Which of the following statements regarding a double helical molecule of DNA is true? Option 1 all hydroxyl groups pentoses are involved in linkages. 2 bases are perpendicular to the axis. 3 each strand is identical. And 4 each strand has parallel 5' to 3' direction. Answer is option 2 bases are perpendicular. Next one. Dash is a multi-locus marker. Option 1 SSR, 2 AFLP, 3 CAPS and 4 RFLP. Answer is option 2 AFLP. Next one. A plant called Rauwolfia serpentina is under the threat of extinction. To save this plant, which technique is highly useful? Option 1 genetic engineering, 2 DNA fingerprinting, 3 hybridoma technology, 4 in vitro culture. 4 in vitro. Next one. A clone is a group of organisms produced by option 1 asexual method and genetically similar, 2 asexual method and genetically dissimilar, 3 sexual method and genetically similar, 4 sexual method and genetically dissimilar. Answer is option 1 asexual method and genetically similar. Next one. Expression vectors are those which option 1 produce protein products, 2 used for genomic libraries, 3 used for chromosome synthesis, 4 used for fingerprinting. Answer is option 1, produce protein products. Next one. Somatic embryos are option 1, embryos developed from zygote after fertilization, 2, embryos developed from egg without fertilization, 3, embryo-like structure developed from cells of callus, and 4, embryos developed by ovules. Answer is option 3, embryo-like structure developed from the cells of callus. Next one. Meristem culture helps in developing option 1, hybrid plant, 2, virus-free plants, 3, disease-resistant plants, 4, tall plants. Answer is option 2, virus. Next one. A part of nucleic acid used to find a gene by hybridization is called option 1, vector, 2, clone, 3, probe, 4, hybrid. Answer is option 3, probe. Next one. A segment of DNA that reads from the same forward and backward is called option 1, palindromic DNA, 2, complementary DNA, 3, plasmid DNA, 4, copy DNA. Answer is option 1, palindromic. Next one. Which one of the following RNA molecule is involved in regulation of gene expression? Option 1, miRNA, 2, 23S RNA, 3, 5S RNA, and 4, tRNA. Answer is option 1, miRNA. Next one. The absence of sigma factor in RNA polymerase. Option 1, affects elongation only, 2, blocks initiation only, 3, affects both initiation and elongation, 4, does not affect transcription. Answer is option 2, blocks initiation only. Next one. The RHT mutation in wheat that were pivotal for green revolution caused reduction in plant height due to impairment in option 1, gibberellic acid biosynthesis pathway, option 2, gibberellic acid signaling pathway, 3, auxin biosynthetic pathway, and 4, auxin response pathway. Answer is option 2, gibberellic acid signaling pathway. Next one. A genotype with an organism with the genotype AABBCCDD is selfed. The 4 genes are independently assorting. What proportion of the progeny will show the genotype? Answer is option 1, 1 by 4. Next one. Pre-germination cold treatment to break dormancy is called option 1, scarification, 2, stratification, 3, dormancy break, 4, vernalization. Answer is option 2, stratification. 1. The number of moles of solute per kilogram of solvent, not solution, is referred to as option 1, molarity, 2, molality, 3, mole fraction, 4, none of these. Answer is option 2, molality. Next one. Tetrasodium chloride commonly used in seed viability testing indicates option 1, cellular respiration, 2, intact cellular and ultrastructure, 3, seed germination, 4, none of these. Answer is option 1, cellular respiration. Next one. The Ames test is used to option 1, detect bacterial viruses, 2, determine the rate of DNA replication, 3, examine the potency of antibiotics, 4, measure the mutagenic effect of various chemical compounds. Answer is option 4, measure the mutagenic effect of various chemical. Next one. The technique used in genome editing is option 1, antisense RNA, 2, site-directed mutagenesis, 3, epigenome modification, 4, CRISPR-Cas9. Answer is option 4, CRISPR-Cas9. Next one. Genomic libraries consist of option 1, fragments of genomic DNA, 2, fragments of cDNA, 3, fragments of RNA, 4, fragments of proteins. Answer is option 1, fragments of genomic DNA. Next one. The number of different types of tRNAs required is less than the number of codon because of option 1, Vobel pairing, 2, triplet codon, 3, unambiguous nature of codon, 4, strict Watson-Crick pairing. Answer is option 1, Vobel pairing. Next one. Transfer of genetic material from one bacterial cell to another by means of virus particles is called option 1, transduction, 2, sexduction, 3, conjugation, 4, transformation. Answer is option 1, transduction. Next one. The exchange of non-sister chromatids of homologous chromosome is known as option 1 reciprocal translocation, 2 recombination, 3 inversion, 4 non-reciprocal translocation. Answer is option 2 recombination. Excuse me. In the B form of DNA, the pentose sugar is dash endo and dash exo deoxy sugar. Option 1 2 prime 3 prime, 2 3 prime 2 prime, 3 2 prime 2 prime, 4 3 prime 3 prime. Answer is option 1 2 prime 3. Next one. The transcription of lac operon is negatively regulated by option 1 repressor, 2 CAMP, 3 lactose, 4 allolactose. Answer is option 1 repressor.